So our next speaker, we're going to start, this is our gallery of BloodWorks Northwest of speakers. We have a long relationship with that institution through Jose Lopez's efforts over the years and collaborations and partnerships. So we're really thrilled to have a whole bunch of speakers from that part of the world, which is similarly rainy. Michelle Lambert is sorry, I'm shifting. Okay, you want to do it again, Michelle? No. Amélie Gros is an associate professor at BloodWorks Research Institute in the Division of Hematology Oncology at the University of Washington. She leads a research program that focuses on immune functions of platelets. She received her medical degree from Shandong University in China and subsequently moved to Toronto for her PhD under the famed John Semple. And then she went on to study with other rock stars in the field, including Andy Weyrich, Matt Brandina, Paul Bray at the University of Utah, with mentorship from Craig, I understand. So we're pleased that she's so close to Seattle, and I'm sure we'll see her lots of times, but today she's going to give a brief presentation. Amélie.
Thanks for the introduction. It's a great honor to be here, especially in the same session with one of my mentors, Dr. Craig Morrell. So my talk is on platelet-mediated CD8 T cell suppression in sepsis. Sepsis is the leading cause of mortality with 20% of global death and no effective treatment. So what is sepsis? It's defined as a severe health condition with organ failures caused by infections.
I was just going to move that arrow out of the way.
So with improved supportive care, as you can see illustrated here, in the early phase, the mortality has dropped, but patients still die with 25 to 50% mortality in 3 years. And a major cause is T cell suppression. So reduced CD8 T cell numbers and impaired functions are a major cause. And the reason is that T cells are long-lived, even up to decades. And it's believed that the acute phase insult of T cells have long-term impact. But whether platelet has a role in this was not clear. So platelet has been recognized as a critical factor in the pathogenesis of sepsis. It's one of the most important, platelet count is one of the most important prognostic marker in sepsis. So patients with lowest platelet count has highest mortality, but they don't often bleed, but rather have thrombotic phenotype. So 20 to 80% of septic patients have detectable thrombosis. And it's believed that during sepsis, it's the activation of platelet by pathogens and immune system that cause significant thrombosis, platelet clearance, as well as platelet interactions with immune cells that further amplifies the immune responses. So as shown here, patients with thrombocytopenia have highest platelet leukocyte aggregates, and that correlates with disease severity. And this illustrates that platelet labeled in red attached to leukocyte in green. So in my postdoc work, we looked at platelet signatures by transcriptome analysis and ribosome footprinting, and shown here, so we analyzed platelets from both septic patient and also septic mice induced by CLP, sickle ligation puncture. So several of those top upregulated pathways direct to MHC class 1 pathway. It's a pathway that's known to regulate CD8 T cell functions. And around the same time, another group published that leukocytes from patients with sepsis and thrombocytopenia have platelet signatures on them. So, for example, PF4, PPBP. And if you look at downregulated pathways of those leukocytes, all of them are T cell related or even T cell specific. So we suspected that this illustrates the classical MHC class 1 pathway. We suspected that platelet can present antigens through MHC class I and regulate CD8 T cell responses. So we first examined if platelet can take up and degrade exogenous antigens. We use a DQ-OVA as a tool, which upon internalization and proteolysis, it triggers a body P fluorescent signal that's detectable. So as you can see here, the body P signal is positive in both platelets from healthy donors and septic patients. And we have consistent findings in our murine model. Next, we examined if the antigen could be loaded to MHC class I and present to the cell surface. So to do that, we used antibody that specifically target one protein peptide in complex with H2KB, which is MHC class I allele. So here platelets are shown in red. You can see under baseline conditions, there's hardly peptide presented by platelets, but under sepsis conditions, there was significant increase in fecal H2KB. This is consistent with findings from Craig earlier. And we also found that this correlates with the expression level of MHC class I, indicates that the expression level of MHC class I is a limiting factor. So next we examined if this synfecal MHC class I can engage T cells in an antigen-specific manner. So the way they interact is that different peptides presented by MHC can engage different TCRs. So we used a T cell that's specific for synfecal H2KB, and we co-cultured with platelet and measured their binding. So not surprisingly, under baseline conditions without the peptide, there are hardly T cells binding. So T cells shown in magenta and platelets in green here. But under septic conditions, as shown on the bottom, you can see there are significant T cells binding to platelets. We also looked at the T cell function by looking at proliferation, and we found that platelets from septic mice could suppress CD8 T cell proliferation. So next we went on to examine whether this has significant impact in vivo. So to do that, we used a beta-2M PL4 model in which the dendritic cells and monocytes have normal MHC class I, but platelet lineage have deficient MHC class I. So when we did the CLP model, we found that knockout mice have protected CD8 T cell population. And summarized here, they have both protected percentage and absolute number. We then looked at antigen-specific responses by giving the mice OVA vaccine before and after sepsis, and look at OVA-specific T cell population. So as shown here, in the knockout group on the right, we found about 5 times more CD8 T cell that's specific for OVA. So our studies suggest that platelet MHC class I are critical in mediating CD8 T cell suppression during sepsis, and the deficiency of MHC class I on platelets showed protective effect. So then we went on to examine if this is conserved in patients, and this is still ongoing work. On the top, it shows the MHC class I expression in platelets. So as you can see in orange, platelets from septic patients do have more MHC class I expression. And when we measured the CD8 T cell population, we did find platelet binding T cells in both naive T cells, that's CD197 and CD45RA positive, as well as in the effector memory CD8 T cell population, that's negative for CD197 and 45RA. We also looked at single-cell RNA-seq data from the CD8 T cells in sepsis. So a small subpopulation of the T cells are positive for platelet markers, highlighted here CD41, GP1B, platelet factor 4, and those are also abundant in platelets. And interestingly, we found one transcript that was downregulated in T cells, and that's RPS4Y1, which is known with stimulating T cell proliferation. So I think it's possible that platelet suppressed T cell suppression via downregulation of this transcript. With that, I'd like to thank the lab members and also Dr. Jose Lopez, who recruited me to BloodWorks, and all my mentors who all supported me for this long journey. Thank you.
MHC class 1, are they coming from the lungs?
That's a great question. So we haven't have enough mice to do a platelet transfusion assay. So we cannot exclude the contribution from megakaryocytes. So one plan we have is to transfuse platelets from wild type and knockout mice to septic mice to look at platelets independent from megs. Yeah.
Thank you.
Great question.
I've probably asked you this before, Emily, but do platelets in sepsis increase other negative regulators of T cells like PD-L1, some of these other negative signals, or is it strictly they're doing something on the T cell side?
Yes. So we have found at least 3 co-inhibitory signals, co-inhibitory ligand on platelet side. So PD-L1 as well as FTH. I think also the other is L-GALS9, but we haven't, we haven't further confirmed that in patient. Yeah.
Okay. So in sepsis, when you have thrombocytopenia, that's associated with worse outcomes, but you would expect thrombocytopenia to be associated with less T cell suppression unless you're only clearing the ones that don't express MHC class 1?
Great question. So I think our studies suggest there are more platelet T cell binding in thrombocytopenia patient. So part of the reason of thrombocytopenia is they're binding to leukocytes and endothelial cells. But if they don't have MHC class 1, as we've seen in the mice, even though they bind, they are not suppressive.